My name is Peter Darch and I'm a professor for biochemistry and human physiology. My company, Darch Scientific, is working since several years in the interdisciplinary topics concerning medicine, biology, biochemistry and physiology. We are working exclusively with cultured cells with in vitro models since many years. In October 2017, Darch Scientific was asked to have a closer look with the methods of this company on the Bicom bioresonance device and to see if there is any possibility to see beneficial effects. And finally, we did it. Although the use of organ-specific cell cultures does not reflect the situation as in a complete organism, as in our body, we have the possibility to show without using experimental animals if there is any effect of this device on the cellular level. It should be mentioned that things are often not so easy as really expected before. We did experiments for several months until we had the solution for the complex model systems, for the complexity of the device, of the Bicom bioresonance device, and we could put the things together. However, we used the program chain pathogen AI for the basis, and we put the subprograms each to 30 minutes so that we had finally 90 minutes of the complete program chain. This chain was used twice and was applied to the cells. The sample tube on the top right side of the Bicom device was filled with the cell culture medium as taken for the cell cultures, because this was the sample which was referred to. One must be aware that the duration of the Bicom device should not exceed 1 hour of exposure. What we did was an exposure time of 180 minutes by using the cells, 2 runs, each run 90 minutes, 180 minutes. But on the other hand, we really wanted to see if the device is possible to have a beneficial effect on the cells. What did we examine? At first, we had a closer look on the cell vitality of cultured connective tissue fibroblasts. And what we really got was that the exposure to the Bicom device and the exposure to the bioresonance really induced the vitality of the connective tissue fibroblasts by approximately 40% in comparison to the untreated control. Based on these first very promising results, we had a closer look on the regeneration, the regenerative potential exactly, of cultured connective tissue fibroblasts. For that purpose, we have seeded the cells into a special silicon frame, and this silicon frame was grown, densed of the cells, and after taking the silicon frame away, we had a very sharp cell-free space in which the cells could migrate and proliferate in order to recolonize this cell-free space. The recolonization process represents the granulation phase of a wound healing process in vivo. What we got was again a stimulation of the recolonization process by use of the bioresonance method, which was stimulated by about 30% when compared to the untreated control. In vivo, the results suggest that there might be a stimulation of the cell regeneration as well as of the wound healing process in our body. 2 years later, in 2020, we investigated the effect of the Bicom bioresonance device on cultivated functional neutrophils. This is a completely other cell type than used before in 2017 or 2018. Neutrophils are the most common type of granulocyte. They play really two roles in our organism. On one hand, they represent part of the innate immune system, which defends the organism against microorganisms, against microbial pathogens which have entered the bloodstream. On the other hand, these cells are also able to migrate through the vessel wall into the inflamed area, into the tissue, and to produce oxygen radicals in this region. So they produce a local oxidative stress. The production of oxygen radicals, mainly superoxide anion radicals, is called oxidative or respiratory burst and is typical for this kind of cell. Although radicals play an essential role in the signal transfer from cell to cell, an excess of the radicals could do a harm to the cells because the natural or the essential enzymes which are responsible for the detoxification of the radicals are overloaded. As a result, the healing process is normally delayed under these conditions. We used the same experimental setup as 2 years before when investigating the connective tissue fibroblasts. The program gene we used was pathogen AI, and it was also used for 180 minutes. We could see that the functional neutrophils were inhibited in their basal cell metabolism by approximately 12% when compared to the untreated controls. This gives us a hint that the bioresonance had a clear effect on the cellular level and reduces the activity of these cells. In accordance to the decrease of the metabolic activity of the functional neutrophils without any stimulation, we found that the production of superoxide anion radicals was also decreased by about 18%. Both results suggest that the reaction chain on cellular and molecular level, which are triggering the further progress of a chronic inflammatory process, might be interrupted in this way, so that the healing of the tissue might occur afterwards. Moreover, the decrease in metabolic activity of the functional neutrophils and in the production of radicals by the functional neutrophils is in a range which is not really relevant for a decrease of the innate immune system. Taken together, a transfer of the cell culture data, as just shown, to the complicated situation in humans with various cellular processes is quite problematic. Nevertheless, the results we got so far indicate that the frequencies of bioresonance are able to have a positive impact on the cells. But more investigations should be done to shed the light more in detail on the different effects of the bioresonance frequencies. Further investigations will bring more clarity into the current knowledge about the bioresonance frequencies and their effect on the cellular level.